Inside your mouth, you have two small, textured lumps of tissue sitting exactly at the back of your throat. For decades, doctors treated the tonsils as an anatomical design flaw, routinely removing them at the first sign of trouble. This illustration documents that early clinical view. Focusing on swelling and pathology, physicians concluded the tissue was disposable. But look at the anatomical cross-sections on the right. Notice their placement. Modern biology recognizes the tonsils sit precisely at the critical junction of the human airway. They act as biological sentinels. Because of this specific anatomical placement, every single breath of air you take and every piece of food you swallow is physically forced to pass directly over their surface. By positioning sensitive outposts in the path of these high-traffic intake valves, your immune system gains an immediate early warning system against external threats. To understand how they intercept those threats, we have to look at this diagram of their microscopic topography. The smooth throat tissue scales down into deep, cavernous ravines called tonsillar crypts. As fluid and air flow rapidly over these crevices, the deliberate, uneven texture radically increases the tissue's overall surface area. Heavier microscopic particles physically fall into these gaps, getting wedged into inescapable dead ends. This microscopic photograph shows the exact result. Notice the dense, tight clumps of rod-shaped bacteria. During routine breathing and eating, common pathogens are drawn in and forcefully aggregated together within these confined spaces. Inside these holding cells, the pathogens are fully alive. They are actively replicating, metabolizing, and dividing, completely unaware that they are caught in a deliberate trap. The microarchitecture relies purely on spatial design to capture targets in the high-velocity environment of the human throat, expending zero energy to make the initial catch. But the trap is only the first step. The walls of these crypts are actually porous borders. Just beneath them, heavily clustered, glowing immune cells wait in the dark. Looking closely at this micrograph, you can see uniform, spiky surface proteins on these viruses. Specialized defender cells reach through the crypt walls to chemically scan the trapped invaders, specifically identifying these exact markers. Once a hostile signature is confirmed, the defender cells instantly begin to multiply, shifting from passive surveillance to an active, localized threat response. The immune cells initiate a live-fire counterattack. They unleash an explosion of highly specific bioluminescent antibodies, engineered specifically to lock onto the unique structure of the trapped invader. The swarm operates with lethal efficiency. The antibodies systematically dismantle the pathogens, breaking apart their outer structures and neutralizing their ability to replicate. By containing this violent biological warfare within an isolated pocket in the throat, the body resolves the infection locally, keeping the rest of the human machine safe from collateral damage. With the immediate threat destroyed, a much longer-term biological strategy begins to unfold. This schematic visualizes immunological memory. The body extracts structural data from the defeated pathogen, absorbing it into a surviving immune cell. These upgraded cells migrate out of the tonsils, traveling through the bloodstream. They distribute rapidly through branching pathways, permanently lighting up distant nodes. This guarantees that if this specific pathogen breaches the body again, the systemic response will be immediate and overwhelming, bypassing initial detection. The human machine is full of these highly engineered, hidden defense mechanisms. To uncover more of them, subscribe to the channel. Ultimately, the localized capture of a single microscopic pathogen by the tonsils is the mechanism that secures the long-term survival of the entire human body.